these sections underneath the microscope. They're little holes in the coloured forms there to the right of the plasmid, the kind of the fraction of algae that you can see on the left. Or was it the other way around with you? And essentially, if you leave a magnetic field underneath a bryopsis culture that is eating magnetite, you can actually re-engineer the form to take up any shape you like. This was just done with a few kitchen magnets to create an Ouroboros, the snake eating its tail, which was kind of suitably alchemical. So essentially, you don't need to be able to manipulate DNA to be able to engineer biology in a kind of a design context. And say, for example, we could use this kind of technique to create synthetic breakwaters. They're actually made of seaweed itself. In other words, it has a seasonal cycle through which deposition and the accumulation of sand on the sea will actually change in a seasonal way. So when the algae is blooming in the summer, it actually stops erosion of the sand on the shore, and during the winter the algae is not there and it allows deposition of the sand. So these kinds of materials can actually have seasonal cycles depending on the life cycle of the organism that's involved. Another one that's being used widely for experimentation is the slime mould. There are also slime mould robots. I work with one of our colleagues is Sagrada, who created a slime mould brain inside a prosthetic robot. This is one of my favourite quotes. It has no functional value for the purpose of this talk, but I just think it's suitably science fiction. So slime moulds are kind of nasty looking, so they really do need proper socialisation and infrastructure. So in the Dallas suburb of Garland, an unidentified growing object, big as a platter, foamy and creamy and pale yellow, terrorised Mrs. Marie Harris's backyard for 3 weeks until it died of sunstroke and nicotine poisoning. So obviously the slime mould needs a bit of a hand when we're thinking about using maybe a giant single cellular organism to create kind of biochemical pulse waves of information exchange in a city context. The materials that I work with mostly are the bottom-up forms of synthetic biology, where I'm using chemistry to generate lifelike interactions. How do I access? No, there's no sound. Essentially, these are just this is an oil and water droplet system. These are protocells. They don't possess any DNA. And as you can see, they undergo lifelike behaviour. In other words, they can sense their environment, they can move around it, they can interact with other agents. And in this case, you're just seeing one shedding a skin and material. So the idea then is to use these agents to create infrastructures that use infrastructures that actually will grow in an urban context. How do I get this back into full speed, please? So the red substance is a microstructure that has been built. Well, let's keep going. Sorry, it's been built by the Protocell system, and essentially the forms are incredibly environmentally contextualized. So if you change the surroundings, you can actually influence the kinds of forms. I can actually tell you the pH of that system is around, I can tell you the molar solution of sodium hydroxide in that, and that's 5 molar, because I can tell by the actual forms that it produces. If it was 6 molar, which is about the strongest you can get, you actually get much more spherical forms. If you get 3 molar, you get much more spirally forms. So you can actually change the molarity and the pH of the surrounding solution and actually induce particular topologies of the microstructures created by the protocell system. We can also make them make deposits. Here we've got an iron-based deposit grown from a culture of protocells. Here these protocells are actually engineered to be about 1 centimetre in diameter. So they're quite large, and these are part of an installation that was at the Venice Biennale, where I was working with Canadian architect Philip Beasley to create a cybernetic environment which sensed the presence of people, responded to environmental changes, and created both digital, physical, and chemical material responses. So in order for these these synthetic biologies to become a reality, we need some manufacturing platforms. Just recently, I've just come back from Glasgow, where we were using a 3D printer to create a wet fabrication lab. So this was with the Cronin Group, Euphoria, a software company, Fab at Home, which makes a printer, and Avatar, which is the research group that I'm working with. So essentially here we're using the olive oil and sodium hydroxide protocell droplet system, and I've put an indicator in there which is copper sulfate. This was really to show where the patches of chemistry were located, and this was really during the calibration process. We also used agar, so we could actually print agar, which contained materials in itself, and actually created a three-dimensional structure which was built with software. So essentially I could distribute chemistry in space and time, use a biopolymer scaffolding, and then actually let the material calculate a response based on time and information and space. In order to scale up then these manufacturing programs, we were working with other forms of expertise. Julius Popp works with where art and science converges, and he's actually expert in the information that resides in systems that deal with wet interfaces. This is actually a set of tubes that you can see in the middle is printing the letter A, and this is an installation called BitFlow, where actually the water is programmed to create form. And here Julius has programmed an interface that is from droplets created from a sprinkler system that actually write letters that have been taken off the internet by a computer program. So there are two forms of data. One is physical data, the other one is data from the internet. So I'm happy about that. So essentially, in order to create a context for the display and for the design within a cityscape, we need to move from the wet to the dry environment. So buildings at the moment are really constructed the same way. They have a concrete and steel skeleton, and they're wrapped by a cladding, which is really a series of plates of glass. The idea is to use the space in these plates of glass to house these wet chemistries, these synthetic biologies which are green goo at the moment. So this is a design for a solar panel that can produce biofuel instead of electricity. This is a bacterial cladding. This is by a colleague, Simon Park, and he's chosen a species of bacteria that draws lines perpendicular to lines of trauma and can also be fed with nitrogenous waste to actually create calcific material. So in other words, if you wrapped a building with a cladding like this, not only would you be able to visibly see where the lines of stress are, but actually at the micro scale, the building, the bacteria on the building would be responding by creating material repair through the deposition of calcite at the microscale. So in other words, bacterial bracing for real-time stresses in buildings without the need of an engineer or a survey. This is a student of mine's work creating a form of algal farm, which is growing a culture around a synthetic speculating on a time in which human and plant matter may be symbiotically interlinked. And essentially these really give rise to a notion of synthetic soils, layers upon layers around our buildings in an ecologically designed way that creates series of interfaces which perform different metabolic functions, different kind of embodied environmental chemistries in the context of the city. So the idea then is that our cities, rather than being sterile deserts, become synthetic ecosystems that are designed with human habitation in mind. So I think there's an important relationship here. Synthetic biology has the potential to create a new kind of material for our cities, a new kind of garden, as it were. But we need the infrastructure to be established in an architectural way that provides the context for technological and social development. And cities are ideally placed for this because actually architecture houses most of our technology, and that's illustrated by its 40% horrific carbon footprint. So architectural engagement in synthetic biology to provide infrastructural support is vital for the innovation and technological and commercial maturation of this particular technology in order for our cities not to be deserts but to be increasingly more lifelike.